In past video, we look after the quantitative and qualitative analysis methods of quality control, whose video is already uploaded on the channel. Now we are going to start with what are the sources of error. Like if we are preparing any kind of product, then there are definitely a chances of error. So how we determine these error and what are the criteria for determining these error and what are actually the error in the product formation. Now we go through with all these. So there are two main classes of error which affect the accuracy and precision of a measured quantity. Accuracy is something which we prepared is completely accurate or not. So that is their accuracy. And if we talk about precision, okay, of a measured quantity. So there are basically two kind of errors. One is called determinate error and second is called indeterminate error. As a name completely indicate determinate whose error or what other things are present in that particular product, we can easily determine that. But in indeterminate error, we cannot identify what contaminants and what other ingredients are present in that kind of product formation. Now we go through one by one. Now, so here, this is a determinate error. Now, these are determinable and can be either avoided or corrected. The error may be constant as in case of, see, the very first important thing is they are determinable. That is most important. They are determinable and either can avoid it or correct it. See, when we correct or avoid anything, when we know that what contaminant or what other ingredient is present in that particular product, then only we can avoid it or even we can correct it. So the error may be constant as in case of weighing with uncalibrated weight, measuring a volume using uncalibrated burette or pipette. Like we can performing an experiment and we cannot calibrate the burette or pipette with the help of chemicals. So in that case, we can easily determine what error should be there. And this can be called a systematic error. And what are the causes of arises? The very first is instrumental error, like if we use uncalibrated equipment during the experiment. Operative error, the by person operating or doing an error, like it is due to the personal error. We cannot take the volume properly. We cannot calibrate the equipment properly. That may be a human error basically. Third is chemical error. Chemical error is due to impurities in chemical, solvent, or reagent, or even if we take the excess or deficient amount of a particular measured chemical. And error in methodology. Error due to unvalidated method. Might be this sometime happen that we cannot go step by step. We perform second step before first step. So that may be an error in methodology. Errors of above categories are usually detectable and can be eliminated to a large extent. Yeah, it can be extended. Like these are a kind of error which we can easily identify if the result is not known. So that's why they are called determinate error. Okay, second is indeterminate error. They're often called accidental or random error. So these are basically what they might be happen randomly, accidentally, and they're found by small differences in series of measurement made by the same analyst under identical conditions, and they can not be predicted. The very important thing, as I already told you that what is the difference between determinate and indeterminate. Determinate can easily be predicted while indeterminate cannot be predicted and hence cannot be eliminated. Obviously, if we should not predict it, then how we can eliminate it? So this is the basic difference between determinate and indeterminate error. Now, what are the sources of impurities in pharmaceutical? Like we talk about determinate, indeterminate error, that there should be impurities in that. Now, what are the sources of impurities? The very first is the substance used in pharmaceutical field should be almost pure. This is the very first. Like we cannot use very pure substance while performing the experiment. That might be one of the impurity. The second is the impurity of a substance vary with different factors, such as their method of manufacturing. Exactly. Method of manufacturing matters. Like how we manufacture any kind of substance, the type of purification, which purification technique we used, what separation technique we use. Like for separation, we use chromatography. Then in chromatography, we look after that there are so many types of chromatography, TLC, HPLC. So what method we use is matter. So that is, might be the second source of impurity. Third, impurity means presence of other material than drug or presence of unwanted foreign particle other than active ingredient. Like if I am preparing any product and I'm having a list of ingredients that these three ingredients is required, but we add there is one more ingredient present, I'm saying fourth one. So this is unwanted foreign particle that we don't want it during the manufacturing, but it is present. So if it is present, then we not get exact result what we want, and we not get the product which we want. So that is basically called impurity. It may be toxic or non-toxic. Exactly, we don't know now what is this food product. It may be toxic, it may be intoxic. Toxic is when like if excess amount of any drug is present, then we call it toxic. And if it is in a deficient amount or having no negative impact on the human body or on the patient or consumer, we can call it non-toxic. It may be used intentionally as adulterant to increase the weight of active ingredient. Sometimes the toxic or non-toxic substances use intentionally as an adulterant to increase the weight of active ingredient. Next, non-toxic impurities also reduce the activity of drug, so that one must avoid impurities in pharmaceutical cannot eliminate all the impurities. And the official pharmacopoeias prescribe limit for particular impurities like sulfate, chloride, iron, heavy metal. These are basically impurities, like there should be sulfate as an ingredient, chloride, iron, heavy metals, arsenic. These may be the impurities which are present in any of the ingredient or in any of the product list. So these are basically a source of impurities. We need to basically take care while we manufacture any product. So we need to take care of all these factors, which is very important for the for getting the perfect result. Now here some factors which are responsible for pharmaceutical impurities. So very first, if we talk about is raw material employed in manufacturing. The very obviously when we start manufacturing any product, as I already told you that the very first basic step is what? Acceptance of raw material. So raw material is very important, or we can say the basic building block for any product formation. So the raw material from which these are prepared often contain impurities. Like I'm just taking an example. When we take the iron ore, so in that iron ore, not only iron is present, it also have other impurities like sulfate, magnesium, aluminium, etc. So in that particular ore, all these impurities are present. But if we need to extract the iron, then we need to delete it out all these impurities from that particular ore. Then it becomes purified. So this is what the raw material from which these are prepared often contain impurities. It is therefore necessary to employ pure chemical and substances raw material. So whatever we are using for the raw material, we need to make sure that that particular raw material is completely pure. So this is very, very important. This purity is very, very important. Just taking an example, presence of tin, lead, silver, copper, cobalt, and gold in bismuth salt. This is a salt which is called bismuth salt. So in that particular salt, other than bismuth, bismuth is also an element. So other than the bismuth, it is having tin, lead, copper, silver, cobalt. So this is not a purified salt. So if we want this purified, then we need to extract it out from these all impurities. Second is rock salt contain a small amount of calcium sulfate and magnesium chloride. If we talk about rock salt, so what is this rock salt? Rock salt is nothing, is just a common salt, which is called NaCl. So this is also called rock salt. So this NaCl is also having a small amount of calcium sulfate as well as magnesium chloride. So these are the two basic impurities which are quite present in a smaller amount, but these are the impurities which is present in what? In our rock salt. So sodium chloride prepared from rock salt with almost contain traces of calcium and magnesium compound and impurity. Exactly. Like even sodium chloride is prepared from this rock salt, as I already told you. So this NaCl is not purified completely. It is having a small amount of impurities of calcium as well as magnesium. So this is the very first factor, like raw material employed in the manufacturing. Second, methods used in the manufacturing. Every pharmaceutical company or any other company is having a different method for manufacturing anything. So here we just look at the basic methods of the manufacturing. Some impurities get incorporated into the material during the manufacturing process. Exactly. Like if we want to enhance the shelf life of any product, to enhance, to add adulterant, then we use some impurities during the manufacturing. So it is completely mentioned. In that, the very first step is intermediate. For certain drug, a multiple step synthesis. See, drug is not a single step procedure that we are having a reactant and we put it into a machine and we get the product. See, this is not a drug manufacturing. Drug manufacturing is a long procedure and a multi-step processor. Here we go through with so many steps. So multi-step synthesis procedure is involved which produce intermediate compound of, say, I am having like this A is a reactant, and now here something is formed, and after that a final product B is formed. So this something, whatever is formed, it is first of all unstable. It's not stable. Okay. So this is unstable. Our product is stable actually. So this unstable product which is formed in the intermediate, which means in moderate between reactant and product, this is called intermediate compound. So the purification of intermediate is essential. If we purify this intermediate, then only we get the purified product. So intermediate purification is important step. Otherwise, impurities present in intermediate will get into the final product. Yeah, obviously, because it is basically a transformation from this is reactant. Here intermediate is formed, and here it is a product. So whatever intermediate is having, they are completely converted into what product. So if intermediate is having impurities, then obviously product should have impurities. That's why if we need a purified product, then we need to purify the intermediate. That is clearly mentioned here. Example is mentioned, potassium iodide is prepared by potassium hydroxide with iodine, and the intermediate potassium iodide formed is reduced to iodine. Like if we are preparing potassium iodide, then for that preparation we require potassium hydroxide with iodine. So if we prepared by this, we are having a intermediate which is potassium iodide. And now if we reduce it, then we get the iodine. So if iodine is not reduced completely, the final product potassium iodide will contain traces of potassium iodide as impurity. Exactly, if the intermediate is not purified properly, then the iodide symptoms also shown on the final product. So this is what if we need a final product should be pure, then we need to purify what? We need to purify intermediate. This is the very first basic method used in the manufacturing. Then second, if we go with the reagent used in the process. Now, what reagents we use in the process? Obviously, for manufacturing anything, we use different kinds of reagent. The final product may contain unreacted reagent. Now here, unreacted reagents. Unreacted reagents means there are some reagents which can be activated only at suitable condition, or we can say a favorable condition. So if these favorable conditions are not present, then they remain unreacted or inactive basically. So they used as an impurities and if not washed properly. Lead is an impurity may result from sulfuric acid used as a reagent. Like when we using sulfuric acid as a reagent, then it always having lead as an impurity. Okay. So this is if we not purified properly. Second is soluble alkyl may be an impurity in calcium carbonate if the calcium carbonate is made by reacting calcium chloride and sodium carbonate. If we mix these two reagents together, calcium chloride and sodium carbonate. So if we mix these two reagents together, then we are having calcium carbonate as an impurity. So this is what impurity. If we not purify this very properly, then the final product which we get should not be purified. So we need to make sure that whatever reagent, whatever intermediate we are using, we need to purify it properly if we want the perfect purified product. Again, the third is solvent. Water is a common solvent in most of the manufacturing in pharmaceuticals. And this can give rise to traces impurities such as sodium. See, there are two kinds of water, tap water and distilled water, which we also call mineral water. Tap water is having maximum mineral content other than water molecules, and distilled is completely purified water, which do not have a single trace of any impurity. So if we use a tap water, then in tap water we are having so many impurities like sodium, calcium, magnesium, carbonate, chloride, sulfate ion. These impurities can be avoided by using purified user. So as you can see in the laboratory, or even you can, whenever you perform experiment in your laboratory, most of the time we prefer water, distilled water, because distilled water is completely purified and it doesn't have the impurities such as sodium, calcium, magnesium, carbonate, chloride, sulfate ion, etc. And when we use distilled water, then chances of error in the result should be avoided. That's why we always prefer distilled water in the lab instead of tap water. Now, second is catalyst. So, first of all, what is the function of catalyst? If we talk about catalyst, what is the function of catalyst and why we use it in any of the reaction? So, the very basic thing is about catalyst is it enhances the rate of reaction. What it does, it enhances the rate of reaction. Enhance means it increase the rate of reaction. What does it do? Increase the rate of reaction, basically. That is called catalyst. Catalysts are used to induce a reaction or to initiate any reaction at a faster rate. This is important. At a faster rate, then we always prefer catalyst. And there may be possibility of incorporation of traces of catalyst in the final product. Maximum time while we use in a chemistry, we always said in whatever form we use catalyst, it come out from that form out of the product. But yeah, traces should be there. So like presence of palladium catalyst in phenanthrene. Whenever we prepare the phenanthrene, we use palladium as a catalyst. So there, like in the product phenanthrene, some traces of palladium also seen. So if we want to purify it, we need to again purify the intermediate, and then presence of copper chloride and synthesis of phenol. Now the reaction vessel. The vessel used in the manufacturing process are made of metal like copper, iron, aluminium, zinc, tin. Though these days many of these metals are replaced by stainless steel. Why they are replaced by stainless steel? Because this stainless steel does not have impurities. That's why we are preferring using stainless steel instead of using the product which is made up of aluminium, zinc, copper, etc. And the chances of contamination reduces if we use a stainless steel. Like glass vessel may give rise to traces of alkyl to the product. If we are using during the experiment glass vessels, then there should be a chance of traces of alkyl found in the product. And metal particles of aluminum containers may contaminate the product like ointment and paste stored in it. So these are the reaction vessels. We need to take care it as well properly. Now, what are the limiting tests? Limiting tests are basically under the category of quantitative tests. As we already studied about what are quantitative tests and what are qualitative tests in the last uploaded video. You can go through with that for knowing the difference between limit tests. Sorry, quantitative and qualitative tests. So limit tests are what? These are quantitative tests. See, what is now limit tests? Limit tests are used to know about impurities. Like if we manufacture any product, then what level of impurities are present in that particular product? To know about these impurities, we perform limit tests. Even these are a tests which are very sensitive and they can detect the small to small impurity either present in the product. That's why we prefer using limit tests. All the limit tests are prescribed in the pharmacopoeias are based on the comparison standard turbidity, color with that sample under test. Yeah, this test is basically based upon the standard turbidity or color of that particular sample under which we are performing. For the preparation of a standard turbidity or color, the pharmacopoeias prescribe the limit of particular impurities for particular substance, and it may vary for different compounds. See, they are specific tests. Like I'm performing test for knowing calcium, then they are having different standard turbidity and color. If I'm performing it for potassium, then they are having different standard turbidity and their color. So it is a very specific type of test which is present for different compounds at a different color and standard turbidities. Usually, the limit tests are prescribed in parts per million. It is again a unit in solution, basically concentration term. This parts per million is concentration term. So it is a concentration terminology. The amount of test sample to be taken is mentioned in individual monograph of the pharmacopoeians. These are the basically limit test. Now, the very first is limit test for chloride. If we want to know that in that particular compound what amount of chloride is present and if the traces of chloride are present or not, then there is some basic test like a solution of a substance is acidified with nitric acid. Like, see, I'm having this solution and I just want to know chloride is present or not. So what should I do very first? I acidify this solution with the help of nitric acid, the very first step. Dilute to definite volume and treat it with silver nitrate. After that, I will treat it with silver nitrate and the opalescene so produced. Opalescene is basically screen color, okay? Opalescene is what? It's basically a color. So produced is compared with that of standard opalescence containing known amount of sodium chloride solution. This is chloride. This is our what silver nitrate. And if we form the product, then silver chloride with nitrate is formed. So presence of nitric acid prevent the precipitation caused by silver carbonate or silver hydroxide, which may result due to alkaline impurities in the solution. So preventing that, if we test such like, then the traces of chloride come out from it. This is the very first step how we can test for the chloride. Okay. Now there is another test, limit test for the sulfate. It depends upon the precipitation of sulfate with barium chloride in presence of hydrochloric acid. Barium chloride. If we want to know the precipitation of sulfate in any of the solution, then we first of all go with the barium chloride. This is BaCl2. In presence of hydrochloric acid, hopefully everybody should know what is hydrochloric acid. This is HCl. Then we go with ethyl alcohol, C2H5OH, and traces of potassium sulfate. So these are the basic chemicals required for the testing the sulfate. The turbidity produced is compared with turbidity produced by addition of above reagent. First of all, we perform this in two samples. One before adding, this is before adding the above reagents, and second test after adding all these reagents. And then we compare. We need to compare the standard turbidity and color before and after. So the potassium sulfate increases the sensitivity of the test by giving ionic concentration to the reagent which just exceed the solubility product of the barium sulfate. See, a potassium sulfate, as I told you, this K2SO4, and if I break it down, it can break into K positive and SO4 to negative. So these are what ionic concentration. Here I talk about ionic concentration. So these are ions. Ions are what? Which are having positive or negative charge, cation and anion. So K is having potassium positive charge, while S is having SO4 is having negative charge. So this provide ionic concentration to the solution, which can help easily to identify the concentration of sulfate in particular solution. The second is presence of alcohol help to prevent supersaturation. We use alcohol here, ethyl alcohol. So what does it do? It prevent supersaturation. And hydrochloric acid is added to prevent precipitation due to barium carbonate. Barium chloride we use, hydrochloric we use, and here you can see SO4 react with BaCl2, it form barium sulfate, which is sparingly soluble in water. So just to prevent its solubility in the water, we use hydrochloric acid. This is the test which is used to know the sulfate concentration in particular solution. Next is limit test for iron. It is a specified amount of drug dissolved in water and treated with citric acid and citric acid is found in what kind of substance? Do you know? In what kind of substance citric acid is found? Let me tell you, it is found in basically sour fruits and vegetables. Sour fruits and vegetables like lemon, orange, etc. And thioglycolic cholic acid, it made alkaline with dilute ammonia solution. If the pure purple color produced is compared with that of standard ferric ammonium sulfate is treated in the same way as a test solution. Here you can see ferric ion reacts with thioglycolic acid, and then ferrous ions react with thioglycolic acid. What do we get? Ferrous thioglycolate complex. This is purple color, and this color indicate that the particular solution is having iron traces. So this is a test how we can test the iron. Ferric iron is reduced to the ferrous iron by thioglycolate. I already told you through the image that what was done there. Now there is a limit test for heavy metals. See, what is this heavy metal? Recently we go with chloride, iron, sulfate. So we mention clearly that what element we are testing. Here we just give a name heavy elements. This is what heavy element. What is this heavy element? They are basically all the elements like copper, bismuth, lead, mercury, arsenic, antimony, silver, etc. except alkyl and alkaline earth metal. In a periodic table, we say group 1 and group 2. So this group 1 is what? Alkyl metal. And this group 2 is what? Alkaline earth metal. These names are given them on the basis of their origination. Okay. So depend upon the quantity of, except these metal, all other metals basically are what? Heavy metals. Depend upon the quantity of metal, the color vary from brown to black. Obviously, if a compound is having little amount of metal, then it is having a brown color, and if it is having a large amount of metal, then it is having a black color. So that's depend on the color. And then three methods are prescribed to determine the presence of heavy metal. Method A and B are carried out in acid condition with hydrogen sulfide reagent. There are three methods prescribed to know about the what content of heavy metal present in particular solution, and that is categorized as method A, B, and C. Method A and B are carried out in acidic condition. Okay, in acidic condition, we react the solution with hydrogen sulfide reagent, and in method C, we react the solution with alkaline medium with sodium sulfide reagent. So here you can see method A. Solution of a substance is adjusted to pH 3 to 4 because as I already told you in method A and B will provide them what? Acidic condition. What will provide them? Acidic condition. So acidic pH 3 and 4 is what? Highly acidic. By adding ammonia or acetic acid, the hydrogen sulfide reagent is mixed with this and comparison of black color produced. See, if after mixing hydrogen sulfide, we are getting a dark black color, then it means it is having a large amount of heavy metal. If it is having a brown color, then it is having a trace of heavy metal. So that is basic difference. For again same for method B because method A and method B are same, only the reagent is different. Here we use sulfuric acid. Method C, the solution substance is treated with the sodium hydroxide solution and sodium sulfide reagent. Then it's compared with a standard color. Again, if it is having a brown color and the result is black color, then we can compare both the colors. One is having brown, another one is having black. So brown is having less traces of heavy metal, while black is having a large traces of heavy metal. So this is a basic difference. Now this is all about basic pharmaceuticals quality control methods. Hopefully it can understand you. Hopefully you can understand all the procedures. Either it is a determinate error, indeterminate error. These are quantitative, qualitative analysis. And if you are having any kind of doubt, just let me know in the comment box. And you can follow the ITLS Academy on Instagram, Facebook, Twitter, YouTube, WhatsApp, LinkedIn. Even you can download the app from the Play Store. Thank you so very much. Like, share, subscribe. Thank you, friends.